So, this is Primary Neural Networks, bombastically arriving to you every Thursday morning. Let's see. So, I hope you had the time to play with multiple of the things that we've discussed last week. And what I'm going to be doing today is continuing, of course, as we started last week in our voyage through neural networks, and particularly biological neural networks and spiking neurons. So I wanted to start out with the outlook of the overall arch, like story arch, that we're having this year in primary neural networks. Here, we started lecture 1 with computational neuroscience versus neural computation, and we already started also introducing the Hodgkin-Huxley model. And you've done a project probably, and you've gone through multiple aspects of that project. I am presuming that the project was relatively low level. I mean, straightforward. I'm hoping we're going to be looking into some specifics. I want to know how the message got across and whether you had fun. I didn't get many questions through Discord for that particular project. We will still be looking into questions for the projects throughout the duration, so you can continuously ask questions about any project at any time. Then lecture 2, this lecture. We had a little bit, I've reconsidered what our material for each day was going to be a little. I didn't want to overburden lecture 2, going from models of spiking neurons all the way to integrate and fire networks. So I've decided to stop at integrate and fire neurons. And then on lecture 3, we're going to be focusing on networks, both spiking networks and non-spiking networks, looking into structure and function relationships. Then on lecture 4, we're going to be looking into the relationships between plasticity and learning. And on lecture 5, we're going to focus on patterns and perception. So precisely how learning bootstraps certain cognitive abilities, such as pattern recognition. And for the final encounter, which is our test day, we're going to have the project presentations. Today, I have released the themes for the projects in Brightspace. So this is the grand story arch, and then particularly for today, we will start out with a review of our Hodgkin and Huxley model and how you've experienced it. I mean, whether the main components of that lecture got across, of that project got across, and that's going to go through a Mentimeter poll in a second. After that, we're going to follow it up with an Hodgkin-Huxley review, which is essential for the lecture today, just to bind the concepts with the project material and create a continuum to what you're going to be looking into today, which is spiking and models of spiking, and our understanding of what are the ideal conditions for spiking and what kinds of spiking neurons can there be as a function of their conductance interplays. So this already happens after the first intermission. So threshold shenanigans, is there really a threshold in the neuron? That's the question for the time after the intermission. And we're going to go into neurodynamics proper, which is the dynamical system study of spiking activity of biological neurons. Then we're going to have an intermission. Later, we're going to look into the reduction of dimensionality. What are the strategies we can have to reduce the dimensionality of the complex Hodgkin and Huxley neurons into more amenable to analysis neurons? The neurodynamics part we're going to have just before is basically giving you the theoretical material that allows you to think about reductions of dimensionality in the context of spiking neurons. I'm going to talk a little bit about parameter spaces, so everything that can happen under the sun. And then for the remainder, we're going to be looking into another project, which is going to treat directly neurodynamics. And then we're going to have questions and answers in Discord. Hope you're hearing me fine. Like no one actually is complaining, so I'm assuming you're having coffee and listening to all of this. And without further ado, I want to bring you to a Mentimeter probe. So here's the code that I would like you to use, 261556. And I wanted you to gauge your own understanding of these fundamentals here. So many of these you have already encountered in previous studies, also during nanobiology, and the project should have bolstered your understanding, filled it up with more content so that you can bring this knowledge forth. So I would like to hear from you. How do you feel about these particular concepts in neural networks? So time constants, activation variables, currents, conductances, capacitance, reversal and resting potential, whether they make sense to you. So very murky, very clear. Then based on your feedback, we're going to be looking a little bit more in detail to each of them in turn. Maybe also it's a good time for you to actually ask a question now. You can use the chat, I think it's most conveniently for this. And you can also ask your question via microphone if you feel so bold. And then we can move forward. So we have already 15 participants. There's a total of 30 people inside this room today. A slightly smaller number. I guess the people also trust that the lectures will be online, but you lose the moment, no? So we want to do this live. Aha. So I am a little bit surprised at the fact that reversal and resting potential is one of the shakiest elements of all of this. I understand on the one hand. I mean, like, so usually if we had more time for this whole course, I would focus on a project 0 that discusses the origins of the reversal potential and the resting potential more closely. In fact, given this feedback that you already had, I think what I'm going to do is I'm going to make it available to you, project 0, so that you can, if you feel shaky about the reversal and the resting potential, which are absolutely fundamental concepts of, cell physiology, I think that it's really good to strengthen that knowledge. And so I'm going to make a little project available that usually we wouldn't deploy, but you can then work through it at your own leisure to buttress this understanding. Now, I also understand, activation variables and time constants, where they actually come from, it's not immediately clear. But on a grand average, you gauge them fairly high. So currents seem to be obvious. The activation variables times the conductances become the currents. So in fact, you do need to understand the activation variables to understand the currents that are coming in the cell, or at least the fluxes. And capacitance seems also to be fine. So basically the rearrangement of the ionic or charges in the cell. And the important thing that I wanted to bring up here for capacitances is the opposite relationship that it has with the leak. It looks similar and equivalent from certain respects, but we have to keep it in mind that the capacitance is acting on the change of membrane potential. So it's basically a dampening at the level of the membrane potential voltage change, whereas the conductances and the currents, the current-driven conductances, ionic conductances, they're coming in the cell, and they don't need, they coming in the cell immediately, and the capacitance basically dampens the rate with which the conductance can change the voltage. hope that this is going to get another up in the slides to follow. So thank you for this. I would like to continue with this quiz. So also, if you have specific questions, is a great time. So to ask in the chat. I'm going to move to the next one. I wanted to also gauge your feeling about, as you start working with Python, you've been thrown, I mean, if you haven't done Python before, you've been thrown in the deep end of the pool immediately. And, oh, that's nice. That's very reassuring when the first one kicks it on to 5, all the end of the spectrum. I can very well imagine that Brian, particularly, is a little mysterious in certain aspects. And this, is okay. But I wanted to share with you this notion of simulator language on top of a programming language, the simulator language. What is it doing in terms of solving these differential equations? So in fact, it's the forward Euler solver. I should actually put here also a question about, have you ever heard about forward Euler or forward method of solving differential equations? These days we're teaching them in computational science ever since this last year. But this is really what Brian is doing in the behind the curtains. It's solving the differential equations via an ODE 4-5 solver, for example, or a forward Euler solver. But this is great. So so basically you feel very secure about how to use Colab. You understand what initialization is doing. You're able to produce the answers by, either by shift enter the code, or by actually, so how I meant the question is in terms of the exercises that you've done. And what a neuron simulator is a little bit murkier. What I'm going to do, given this, and I'm going to note it down for myself, I'm going to run a 5-minute short snippet video explaining what the neural simulator does and how it ties in with Brian. I'm going to do this offline. I just wanted to have this feedback first, and so appreciate it. Now we have 22 responders out of 30 participants. We have 25 in the one before. So I think that that's okay. I will move to the next slide if you don't mind. And this is about the exercises themselves. So I've, particularly part 2 of the project, I don't actually even know if you went all the way to part 2. I would be happy if you went until the end of part 1, just executing the code that was already there and trying to understand it. That's actually more than enough for the purposes of the course. But nevertheless, I wanted to know how compelled you felt to go through this. Okay, that's not too bad. I mean, like enjoying debugging is a tough one, isn't it? So I absolutely understand. So also for people that didn't know Python before, I imagine that producing new code would have been a challenge, and we were not so expected. Maybe copying parts that would have been useful. I mean, to get a hang of it. But I imagine that this 1/3 of people that actually produced new code were the people who were a little bit more confident already with Python. For those of you, you haven't actually asked much in Discord, so it seems to be very self-explanatory. Brian, too, as a simulator, has many points of failure, and it's particularly difficult to debug because of the 2 levels. One is the programming level, and the other one is the consistency of the equations level. Units become very important to Brian, and particularly that the state variables are all there wrapped inside your neural group correctly. And so it gives messages that might be particularly difficult to debug if you weren't given the units ahead of time. If you're designing your own model, this is one of the possible pains that the brain involves. On the other hand, by having this strict requirement that the units should be consistent in the system of units, international system of units, you get a very physically meaningful model. And this is incredibly valuable at that level. So you are dealing with state variables that very closely reflect what's going in the cell. And so it's understandable that you might struggle to get Brian's models working, and this maybe also relates to the project that we're going to be deploying today. But on the other hand, I wanted to give you the view that this is very useful to think about. I mean, to lock in those variables and units as particular representatives of this kind of modeling we do. So thank you very much for your feedback. So most of you read and understood the code, produced 1/3 produced code. That's kind of like what I had in mind. It's always good. The code ran in the end. So 50%, that means that some people tried and failed. Come back to us. I mean, like copy some error messages, dump them some screenshots with feedbacks from Brian, and we will be able to help you as well. If you want to do a little bit of more Hodgkin-Huxley problem today, you're also welcome to. Okay, so this is then 15 minutes for this part of the feedback. I would like to quickly open it again for questions. And I have to always bring the chat to this page I'm handling here. And I wanted to now review the Hodgkin-Huxley, the most important concepts, maybe tying back to what your insecurities about it may have been, particularly activation variables, because those are very central to the understanding of Hodgkin-Huxley's, not only, but conductance-based models. Hodgkin-Huxley is the mothership of a great class of biophysically meaningful conductance-based models. So the executive message here is that the current going through the circuit is the sum of the individual ionic currents. And as you've, one of the things that I wanted to home in here today in this equation is the number of dimensions, effectively, of this model. So when we speak about dimension of a dynamical systems model, and we're going to come back to that, we're really talking about the state variables. And the state variables are the ones that indicate what's the current state of the model. For example, the reversal potentials are not state variables. They're parameters. They're things that happen in the cell. They have to do with the concentrations, but we imagine them as being largely constant during the action potential or during the activity. They can change, and there are actually models that make them change, but it's a solid simplification to imagine that they are actually fairly constant. So they're parameters, they're not variables. Then you have the maximum conductances, such as G bar, GK, GL, G bar K, G bar sodium, G bar leak. Those are maximum values representing the maximum number of ion channels. Those are also parameters. But if you want to know what your neuron is like right now, given those parameters, you need to specify, in the case of Hodgkin-Huxley, 4 numbers. I should actually have asked this as a question in the group chat, but I think that you kind of figure this out. Think about it for a little moment. So which variables would you need to specify to actually know what is the state of the entire state of the neural network of the neuron model you have. So you can calculate the difference of voltage and membrane potential if you have knowledge about what's the proportion of sodium channels that are open, what's the proportion of potassium channels that are open, and what's the proportion of sodium channels that are inactivated. So N, M, and H are three fundamental variables that indicate the state of your neuron. And V would be the master state variable, the one that requires all of the activation variables to basically be computed. So the four state variables here would be V, M, N, and H. The differential equations we have, V dot, N dot, M dot, and H dot, tell us how they change over time. So if you want to know the state in the future, what you need is the state of the moment plus the rule of how the state changes. We're going to be looking into this from the neurodynamics perspective in the second half of the class. But for the moment, save the number. Hodgkin Huxley has 4 state variables. The simplification of the equation, you might have wondered about the negative sign. There is a comment in the project about this, so go back to it. We're going to actually also go a little bit more into it. But in principle, the capacitive current, which is a function of the differential of the membrane potential, plus all of the conductances should be equal to the overall current coming through the circuit. And here we have the GI. This is actually a function for every single ion. So I here is the ion. So it could be sodium, could be potassium. It could be many other kinds of ions. And regulated by the particular activation mechanisms that we imagine in the ion channels. So here the sodium channel having both an opening and closing, M, activation variable that goes from 0 and 1, so proportion of channels open or close, and H, the actual inactivation variable. M has a exponent of 3. You said you understood most of you understood the activation variables, but this is something that actually comes up often as a doubt. Why the cube? So there are multiple reasons for that with different levels of analysis. One is, first and foremost, because Hodgkin and Huxley needed the cube in the equations to fit the action potential well. So that's number one reason, and it's basically a fitting reason. So when they fitted it, or the equations originally, they've discovered that if they had the cube on the activation variable for M, the onset of the action potential was faster in the model. So it was convenient to have a exponent there to make sure that the rise is fast, goes from 0 to 1 very quickly. Now this is the pragmatic reason, if you like, so the fitting related reason. But more interestingly, it turns out that this cube relates to the number of degrees of freedom of the sodium channel, of the squid in this case. So you have 4 subunits, and one subunit is the inactivating subunit. That one doesn't move, but the other 3 can change their conformations. And in order for the sodium channel to be letting sodium ions through, you need the 3 subunits to be in the open conformation. So basically, if you think about M, the activation variable, as being the probability of a single subunit, if you have the 3 subunits independently moving, probability would suggest that in order for you to have the channel open, you will have to have m times m times m, so m cubed. And similarly to the potassium channel, m to the power 4th. So that actually is a prediction of the Hodgkin-Huxley about what the structure of the ion channel is. And that's what's kind of mind-blowing, that after fitting this the channel very closely, Hodgkin and Huxley come up with the discovery, still to be done like 40 years in the future, after crystallography of sodium channels, that this exponent relates to a particular structure. So this is obviously a very powerful model prediction. And that's, you know, one of the many reasons why they're so deserving of the Nobel Prize for physiology back in the day, because these predictions are embedded in the Hodgkin-Huxley model. So the exponents here then relating to degrees of freedom generally, but also obtained via fitting often. We haven't talked much about the experiments that are used to measure these activation variables. So where does these cubes come from? And many of them come from experiments such as the ones that I've asked you to do in the project. For example, you can give a holding potential, giving a holding current, and see what happens at the membrane potential. With in the back of your mind, thinking that whatever happened in the membrane potential must have to do with the state of the activation variables. M is related to the sodium. M is related to the steady state of M, and the steady state of M is related to the membrane potential. So first you change the membrane potential, then the steady state moves to a different position. So for example, so if you give a very positive membrane potential to the cell with a current clamp, then very quickly the steady state changes immediately. It says, okay, we want to be open because we are in a very high membrane potential, and so sodium subunits all get their open conformation, making M close to 1 in the steady state. But until we get there, we still have to open the channels. So we know that the steady state at that membrane potential is a certain current. You obtain this by looking at the relationship between the current injected, the voltage that you have, and what is the new membrane potential. So after choosing a holding potential, you see how many currents, sodium currents, came in, like via the fast deflections here. For example, I don't know if you can see my cursor, I hope you can, but when you change the stepping potential in this voltage clamp, you have a fast and a slow component. The slow components relating to the steady state, and fast component relating to the transient. And so you make these experiments multiple times in the cell, and you gather values for your proportion of channels open, imagining that if you have a very high potential, you have maximally open, you have a very low potential, maximally closed. Things in between relate the current to the activation variable. Hope that was clear. So these are the experiments that one would do to, oh, thank you for the feedback, yes. So this is the experiment then that you would do to measure M. You can inspect this slide closely. It's also figure 2.10 in Izhikevich's book. But basically, you relate the activation to the voltage current relationship. So it's so-called the IV curve. So what you see here in the panel indicated by the cursor is the IV current. So you give a membrane voltage via a voltage clamp to the cell, and you measure the current that is necessary to keep that holding potential. But by that, you can then infer what is the proportion of channels open. And so you obtain your M activation variable that looks like a sigmoid from experiment. And that's why we use the sigmoid also to represent it. And so here we also have a tau. You may have noticed that the taus for sodium are incredibly fast. So the time that it takes for the sodium channels to open after a voltage deflection is very small. So if you see tau here is at level of 1.2 milliseconds. That's experimental data. You see actually the error bars. The fact that the error bars are squashed here with very high voltage holdings is the fact that it's so fast changing the membrane, the changing of the membrane potential to the steady variable that it's almost negligible. You see, and really so the model that I presented to you of the Hodgkin-Huxley is not strictly the one that has been proposed by them. I prefer going for a model that has a little bit more inherent simplicity and generality about it. So I chose to present you with a model where the activation variables are sigmoids and the tau variables are Gaussians. And the reason for this should be somewhat intuitive. Not only it comes from experiment, but also in the case of the sigmoid, you go from all channels closed to all channels open, but you go smoothly, right? So as a function of V. Now the tau, there is a maximum duration that it takes for your channels to open and close, and that one is at the place where you are right in the middle of your sigmoidal curve. There's a relationship between the two. Actually, that's not a necessary condition, but for intuition, it's sufficient. It's important to then look at it. So to understand really the Hodgkin-Huxley is to understand the activation variables. And here we have the steady states where we can use our mind's eye to imagine what would be the actual state of the Hodgkin-Huxley momentarily if the voltage deflection was changed. So say here we are at where my cursor is pointing. So notice that these activation variables here are the shifted potentials from Hodgkin-Huxley, minus 65 millivolts they've added to the resting membrane potential because it made their analysis easier. But you have to remember that it's actually the resting potential is these values minus 65 millivolts. Okay. Anyways, so if you're here at where my cursor is, you can see that you have some open channels for M. You have most of the channels deactivated. So you have a little bit of sodium current available coming in, making the cell depolarize. But you also have open potassium channels, a certain proportion. And they're balancing each other out. So this is what's called a window current. So sodium is constantly coming in, potassium is constantly going out. And so there is balance between these two currents because, have open potassium, open sodium. They're balancing each other out. In the back of your minds, you should also remember that there's the sodium potassium pump that is actually equalizing the concentrations in the cell. But if you're thinking about electrical currents exclusively, you're thinking about this so-called window current here. So there is a point in the Hodgkin-Huxley where a certain current is constantly coming in and a certain current is constantly coming out. But V dot, that is dV dt, that is the change of membrane potential, is 0 because they're balanced. So this is the window current. I wanted to add this slide particularly so that when you're revising this, you can see what you can remind yourself that during these interval, you have a window current. Now back into the degeneration mechanisms. This should make a lot of sense now, and I think that the majority of you already did, but I get back to it. Hope that you're able to understand all of these subcomponents. They are all equally explanatory. I'm not going to go into this because the project was about this. Hope that you've done the initial part 2 of the Hodgkin-Huxley project. It's reproducing this figure, and so can get even more intuition by looking into the interchanges between H, N, and M. Now, some of you may have noticed that in part 2 of Hodgkin-Huxley project, in one sub-exercise, I have specified some values for M, N, and H, and V with many numbers after the comma. And you might have wondered, where do these numbers come from? So, a quick explanation. Brian starts the state variables, if you do not initialize them, with zeros. But if you initialize the model with zeros, the model has to relax until it's equilibrium. And so if you start, for example, with 0 sodium current coming in and 0 potassium coming out, and you start with a 0 membrane potential value, you actually have to go from 0 all the way to minus 70 millivolts, because that's where the resting membrane potential will be. And so that will create a transient. So you will see like a little bump that you might have noticed in some of the earlier simulations. And that bump is called a transient. It's the time for the time series to get to its equilibrium state. And in Brian, you can save that state, or you can observe that state and then use it later on, so as to have a start for your model that is already at a resting state, so that you don't need to waste that computational time for the transient. And so that's also why here you can see that the gating variables have initial values, and those are at the equilibrium. So the values that I have are the equilibrium values. One more way to look into this, the conductances for sodium. So the resting membrane potential is effectively a weighted average of the conductances and the reversal potentials, as you can see here, Vrest. Now, when you have your membrane potential here where the gray arrow is, you see that as a low, very low membrane potential value, the sodium current is very small. So the weight of it, there are very few sodium channels open. If you flick that membrane potential quickly to a positive value, the sodium conductance grows because the activation variable sodium opens, and your new resting membrane potential is very positive. So your cell, once you change your potential, will have a new powerful sodium conductance, at least during the action potential. This is just before the inactivation variables kick in. So you then have a very powerful conductance, which shifts the whole resting potential. And that's actually what pushes the membrane potential during the action potential to very depolarized positive values. Cool, right? So it's an interesting way to think about this. This is a fulcrum. So here the metaphor is that you have a lever, and the Vrest is basically a fulcrum, and you apply some weights on these fulcrum to set where the equilibrium would be. So I think it's a very powerful metaphor for the resting membrane potential during action potential. So here's rest, here's action potential. And ultimately, the action potential is made of these two positive and negative feedbacks. if you did not have inactivation variables of sodium, then once you inject a little bit of voltage, sodium channels open, and that makes the sodium, it makes the resting potential grow. Like so, our new membrane potential a little bit higher. You continue to add current via sodium channels that are opening. So more channels open, the more voltage, the more channels open. So that's the positive feedback here on the left. If you did not have the sodium, potassium or inactivation channels, then that would actually take the resting potential to this new value here, very elevated at 60 millivolts, close to the sodium reversal potential, and stay there. So the action potential will actually just be a toggle, which would go from low resting potential to a higher resting potential if there were only sodium channels. But since there are potassium channels that activate with a high voltage and inactivation channels that activate with a high voltage, you actually push the resting membrane potential again down. And that's what causes the refractory period and the hyperpolarization or repolarization of the neuron. Okay, so action potential then is a mixture of these two things in the Hodgkin-Huxley. We only have three currents: potassium, sodium, and leak. Maybe other current you were injecting. But I wanted to give you a sense of how many kinds of channels and their actions can be, and where they are located. So if you're thinking about a complex neuronal model, people have been measuring, the activation variables for different kinds of ion channels, and this is what they find. So we have this is on the x-axis. You have the half voltage of the sigmoid, basically where is the middle point of the activation. So you can see that, for example, potassium, our main potassium current here, IK, is activated at a high threshold and has a fairly slow time constant. We also have then the difference between the rectangles, which are the activation variables, and the inactivations of the currents, which are the ellipses. So this is just a sample of the kinds of ion channels that we can have inside the system. So the trends and currents then are the arrows. And so this wraps together what I wanted to tell you about the Hodgkin-Huxley. So now we have 9:45. We started at 9. We're a little bit delayed, so I want to give you a 10-minute oversight of what compartmental models are before we go into the next step of our lecture. So the Hodgkin-Huxley model is a bubble, and in that bubble you have ion channels around it, and this is a very good representation of a cylindrical squid axon. But obviously you know that neurons have dendritic trees, have axons. How do these currents relate to each other is a question. Had a little bit of an animated GIF at the end of the project to give you a view of how saltatory conductance works like, and now I want to give a couple more theoretical angles so that it can help think about these. And also I'm going to give you a couple of empirical results of using compartmental models instead of what are called single compartment models. So the Hodgkin-Huxley original is a single compartment model. We can easily extend it into a multi-compartmental model. So in the compartmental model, we see the neuron as thousands of small cylinders. This cell is a Purkinje cell from the cerebellum reconstructed from anatomy. So people first traced the cell in the microscope, and then they measure the diameters, and they approximate the complex morphology of the cell, which kind of wobbles, with these cylinders. It's a very good approximation at first, and you can actually show this. By modeling the cell as many compartments, you introduce some boundary conditions. For example, the dendrite has a tip, and so at the tip, if the dendrite is very small, then there is no current coming out of it. Shape matters quite a bit to the activity of the cell. So a cell with the same densities of ion channels will actually act very differently. So reconstructions become a very powerful way to look into the class of what a certain type of cell can do. And another thing that the compartmental model allows you is to model the different compartments with different ion channel densities or distributions. Just let me focus on this last topic quickly. You have G bar for sodium, for example, and that represents, in the Hodgkin-Huxley model as we had, millisiemens per centimeter square. Why centimeter doesn't matter, could be in micrometer, of course, but anyways, it's a density. It's a number of channels per unit surface. So that applies generally for any kind of compartment. So you can, for example, have a constant maximal conductance for all compartments, because after all, they're all made of membrane, and the membrane has the same available number of spots for the channels. But if you know a little bit of biology, you know that things are never that simple. And so what actually tends to happen is that axons, somata, and dendrites might express different kinds of channels. Particularly in the soma, you have a lot of sodium channels, and even more so in the axon hillock, in the first initial segment of the axon. You have a lot of sodium ions, and that's where the action potential in most cells is generated, because that's the place where the maximum positive feedback takes place, lovingly. And in the dendrites, you have other channels. Maybe you have a current that is hyperpolarization activated. So compartmental models allow you to have a description of the cell that takes into account the differential expression of densities of ion channels along this morphology. And these interchanges are very fundamental to certain activities of cells. The cell, one of the cells that I study in the inferior olive, has very fundamental differences between soma and somata and dendrites, and I will show you a nice video of them next lecture. I'll come back to this point. But here are the things that might come completely unexpected to you and are results of compartmental modeling. For example, on A here, you have a compartmental model that has been given a steady current. And that's a single neuron that has been given a steady current multiple times, one after the other. And as you see, there is quite a bit of variability in the different traces. So every single spike train, like they're same cell, multiple trials. And so the trials are diverging. They produce the same spike more or less at the same time, but as time goes on, they start to drift apart. So there's a lot of variability in what the neuron is doing if you're giving him a constant, if you're giving it a constant current. Surprising fact appears when you actually inject. So you could see, well, neurons are not reliable. They are biological things, and then they do every time different things, and the variability is enormous. So there's a lot of entropy. In comes B, our panel B. And in panel B, we also have multiple trials. Only that now it seems like it's only a single trial because they're so perfectly aligned. What we've done now is we've injected current that is a noisy current, frozen noise as we call it. So the same fluctuating current every time with the same charge as being delivered by this step in A. And you see now that that noisy current creates spikes at the right time most of the trials. There's a couple trials that do something a little bit different here, but for the most trials, you have almost exactly the same spike times. Very reliable. This is a result from 1995, and it has been reproduced by a compartmental model of the pyramidal cell very closely. This actually, in the Hodgkin-Huxley, is harder to reproduce. Another interesting consequence by the same group, Mainen and Sejnowski, they had like one Science and one Nature paper 1 year after the other. Simple modeling work. They're just, you know, how nice it is to be at the crisp of the zeitgeist wave. So what they showed here is that a neuron with the exact same sodium, potassium densities in the different parts of the cell, these are different cells, A, B, C, and D, all of them have the identical densities for the conductances. So the value for G bar for every single compartment is identical. And so by changing the cell morphology, these are reconstructed cells, you can see that they are doing something completely different. So the first cell is irregularly firing, the second cell is adapting in the lower rate, the third cell chatters because it produces a bunch of spikes and then it silences, and even more so in D, which is a pyramidal cell in the human cortex. Or cat cortex, I think, in this case, maybe. And so important to think about how morphology might influence, even though you have the same parameters for all of these cells, same parameters. The only thing that is varying is the morphology. You have this fundamental difference. How do you get to that? Well, the trick here is that you're basically cutting the cell in the cylinders, as I mentioned before. And these little compartments that represent the dendrites in a certain way, they have, so each of these cylinder pieces, here, for example, in B, this guy, sends current to the guy above and sends current to the guy below and receives from their neighbors too. And this is how we model it. We model it via these axial resistances here. Do you see RA? So RA is the component that is coming from the neighboring compartments. You can also model this as a second order differential equation, but this is already discretized in space. I don't think it's worthwhile to go into the actual derivation of the second order differential equation. It's much easier to think about already discretized. And so here in the bottom equation, what you get is on top of your usual dynamics from the currents, here the sum of currents in K, these are the different ion currents. You have the contributions of the neighboring compartments. So in R prime A and RA, so these are the axial resistances, and V prime M minus VM. VM is us, and V prime is the guy to the right. I don't know if I have actually one of the videos in front of you. So V prime is to the right, and V double prime is to the left. And so these are the contributions that are coming from a radial axial. It's going along the axial dimension with the resistance, and that resistance is a known parameter. So in principle, we are actually just simply assuming that there's conservation of charge. What comes in, what comes out. So if you have here now a discretized axon into many compartments J, and you have then time in T, if you start an action potential at the first compartment, J1, that propagates via charge to all of the connected compartments without loss. At least at the radial dimension. It could very well be that you do get leak across the outward membrane, but not within the neuron. And so you see here on the left panel, you see the 50th compartment and the 70th compartment, and you see that it took about 20 milliseconds for them to propagate from one to the other. This is just a demonstration of conservation of charge in compartmental modeling, which is the basic assumption underlying what we just saw in the previous slide. And the differences in compartmental modeling I had already mentioned, but here is an interesting one in terms of synaptic integration. So this is a granular cell of the cerebellum. And here we talk about synapses. These granule cells receiving an inhibitory and excitatory currents from synapses in the same compartment or in different dendritic compartments. And we plot the activity at the somata of the cell. And here what we see is that if you excite and inhibit at the same time, but in different dendrites, in this particular cell model, you actually produce a spike, but you do not if inhibition and excitation coincide in the same dendrite. So this gives yet another free dimension for the, or another degree of freedom, a complex degree of freedom for the synaptic interactions at the level of dendritic trees. Okay, so flyby, I showed you this in the first, now you have much more material to comprehend what was done in the reconstruction and simulation of neocortical microcircuitry. We've done a lot of compartmental models that were based on reconstructions with differential expression of different conductances in axons and dendrites, blue and red, here in different layers of the cortex, both for excitatory and inhibitory cells. So these are the cells from the somatosensory cortex of the mouse that have been reconstructed to emulate the activity. And this picture shows then on the left the cells in this column that had been reconstructed. In B is a spike raster for each for the different cells in the different layers. In C, you can see the action potentials themselves with some chattering neurons, some regularly firing neurons. And on D, you see probability of spike counts. We're going to be looking a little bit more into spike counts of networks in our next lecture as well. Okay. 9:56. I've overstretched the first part of this for 10 minutes. So my suggestion is now that we stop for how many minutes did I say? 15. So shall we do? Let's do 10:10. Is that good? So we meet here at 10:10 again. And I take questions. I take questions meanwhile. So for anyone that wants to stay, anyone that needs a physiological pause, we can also discuss your questions as I'm here. Enjoy your coffee. All right. So my next slide is this. Oh, neuron, pray tell, what makes you spike? And the thing is that although the spiking mechanism of the Hodgkin-Huxley might give rise for our thought that it's simply some sort of threshold, you need to have a certain amount of current to bring the neuron to the positive feedback region of the positive feedback region of the sodium channel particularly. In fact, the whole story is a bit more complicated, isn't it? So what we observe in reality is that there is a number of different situations that do not act as straightforwardly as this representation here. So in this representation, clearly, the spikes are all or none. You have given maybe current inputs or maybe synaptic inputs to the cell. You brought it from the resting state to, with incrementally growing currents. The ones that are not sufficient don't spike. The ones that are sufficient do spike. So that's the most simple conception of what causes a spike. Oh, were the reality to be so simple? Wouldn't it be nice? Problems. The problems is the threshold is not such a well-defined concept for most neurons. And that has to do with the interplay between the different conductances, the dynamic interplay between the different conductances. So here we have one set of weird cases that make you think about the ways in which a simple threshold are not a good representation of what actually happens in the neuron. In A, we have injected pulses of different amplitudes. And what you see is, at the onset of the current, you have all of them go for a little positive deflection, and there's some sort of like a spikelet kind of thing. But the real spikes actually come later. And if you attend to the individual traces, you will notice that it is not the subthreshold response that determines whether there's going to be a spike, and the spikes happen in different places. So basically, it's not a simple function that just cross threshold spike. No, it crossed the threshold early, but it only spiked late. That's murky. Now we go to B. You have injected pulses of current of different amplitudes, and all of the currents spike. Only later. The lower current steps lead the cell to spike later, but they eventually spike. So how do you relate something that has this continuous relationship here between an inject current and spike? And there is no all or none here, because it's more about when it happened than whether it happened. It will happen at any current, only that it will happen later and later and later. In C, we don't even know. I mean, that's actually a real recording. Actually, it's a real recording of in vitro of a pyramidal layer of 2-3 rats visual cortex. And then you see an example of the modeling just above. I mean, so you start spiking later and later, but you always do spike. And in the B on the side, you also have a funny spiking behavior, because the spike shapes are not infinitely fast. So at changing current, the shapes change continuously. So there is no really well-defined current. And this is actually also explainable via the dynamical systems interpretation of the neuron. This is, in this case, a model. But this is a common behavior also in vitro recordings. So, and then there's just the ultimate fun complexity here. You give to the neuron a negative current, and then when you release, it spikes. So basically, it's a rebound spike. So do you have a negative current threshold? So how can you have a threshold if actually you're injected negative current? Hope that this small set of conundra gives you the flavor of what is the question that we have in mind. What determines the neuronal threshold? And what determines the neuronal threshold is not particularly clear. So there is no real relationship. You cannot derive for most neurons a relationship between its conductances and a threshold. Try to find, the question in maths would look like, what's theta? What's the theta function threshold given a certain Hodgkin-Huxley neuron? Maybe you can derive it if you make certain assumptions about the exact current injection shapes, but you cannot derive it in general, right? And this is yet another way to look into the problem. Here we have a Hodgkin and Huxley neuron, and what we're doing is we're injecting current from one plateau, I1, to another plateau, I2. And we are plotting in Q, S, and R here different kinds of behaviors given the I1 and I2 in the X and Y axis. So in Y-axis, you see I1. In X-axis, you see I2. Q represents quiescent neurons. So you see here that this dot has a neuron that didn't spike. S represents a single spike. So this dot here represents a single spike. And R represents a regularly firing neuron. What this plot here is showing is that there is no single current step that relates to spiking or not. And in fact, the relationship is nonlinear. What divides Q from S is not some sort of straight line. And so that's why you cannot find a single current potential that is the so-called rail base, or the threshold current. Rail base is how it's used to be called. I'm going to type it down. Rail base. No one calls it like that anymore, but or people still call it like that, but it's a historical term. It used to be a scientific question. What's the current that causes a neuron to spike? The rail base. What's the rail base? There isn't one proper because of these complexities. And so so I want to give you a flavor of everywhere we can go with spiking now. You already saw some neurons that spike regularly, some neurons that produce single spikes. And after the ion channel zoo that I've showed you earlier with these different kinetic conductances, we have a spiking bestiary. Turns out, this is chapter 5 in Dynamical Systems in Neuroscience. Fantastic book, like really established. Okay, couldn't you call that nonlinear line a threshold rail base, just more complexly defined? Well, unfortunately, even that example is overly simplified. So maybe for particular neurons, you could have even a linear threshold, and in fact, integrate and fire neurons will define a specific threshold. Only that we have to keep in mind that that only applies to a very small subset of all number of cells. So that was the question by Sarah Okhausen. And I think it's a fair question. I have also, asked my teacher the same question the first time that I was exposed to this. In that particular case of that Hodgkin-Huxley, you could argue that you have a function for the real base. Only that it depends. It's a fairly simple neuron where we were doing it for. It's just a representation of where the complexity starts. I hope that has answered your question. So because you can have more conductances, maybe for a single neuron, you can sometimes define the threshold, but in general, you cannot. Or in fact, you have to think about it more complexly. So if you define the rail base as the amount of current, then that varies a lot with all of the others. So the system is so complex, we don't know the response function. Well, that's also not quite true. So the response function is unique for different kinds of systems. And so we are able to measure and derive what this response function will be, the spikes themselves. But we cannot have one. No? So maybe it's a set of response functions. In fact, let me move forward and hope that this is going to give a little bit deeper understanding of what we're all on. Turns out that, very broadly speaking, actually, let me start this differently. Let's have a chat, a Zoom chat moment here. What differences can you spot between neuron 1 on the left and neuron 2 on the right? So let's start our discussion with a phenomenological discussion. You have a game of 7 differences here, and you have to find the 7 differences between the left and the right figure. Can you do that? Can you contribute with me, like what you think is different between the two traces? The amplitude of the injected current is fundamentally different. A very good point. The initial potential is earlier in type 1 on the left. The spiking frequency, if you can specify what you mean by spiking frequency, do you mean that the one neuron is firing faster than the other, or do you talk about the modulation of the spiking frequency with the amplitude of the ramp? The delay in transition into, you have your eyes very well attuned to the important differences here. When I present this to different classes, I get more simpler answers. So you're spotting the fundamental aspects that are different here. Indeed. So there's a delay in the transition of 2, despite the fact that the current injected is much higher. So you have a rise of the membrane potential, but the onset of spiking is fairly sharp. And after you start spiking, the modulation of frequency on the right side is fairly small. So the frequency of increase is really small. Also, peak of the spike, great. So the peak of the spike on the right changes with the current injection, but the frequency does not. Exactly, the envelope of the amplitude. So the peak of the spike, that's what I took it to mean. Whereas the peak of the spike in the left side was more or less constant. So great observations. Thank you for this. These are the essences of the differences between type 1 and type 2. Let me actually read the question by Renske. I thought that one neuron fired more spikes per second, but that may not be the case because I now see that the time axis is a somewhat different scaling. Yeah, but the important thing here is Renske, that the right side modulates less, even though the current difference is larger in the ramp. Whereas on the type 1, it's more adaptable, it's more flexible. So there's a slow firing rate at low currents and high firing rate at high currents. There's one thing that you haven't pointed out that I want to point out. So these spikes go back into the past, apparently, right? So this probably is some sort of a vector graphics export problem in the original book. You can contribute as an erratum. So this is a spike goes back to the past. No, sorry, but I hope that these got clear. So I think that you've nailed it. This is what there is. And we then talk about, these have been spotted originally even by Hodgkin and Huxley themselves. So when they were researching different kinds of neurons, and they discussed the class 1 neural excitability, where action potentials can be generated with arbitrarily low frequency depending on the strength of the applied current, and class 2, where the action potentials are generated in a certain frequency that is relatively insensitive to changes in the strength of the applied current. And now we understand dynamically why these differences occur. Now you also have here the different types. So brainstem cell is a robustly firing cell that has a toggle. Basically, you give it a frequency and then it starts spontaneously sustained firing at more or less same frequency, driving things. Think about a pump in the back of your mind. It's pumping energy to the cortex. On the left side, you have the layer 5 pyramidal cell. You have a graded response. You have some sort of encoding of the current amplitude. So on the left, you have a good encoding of current. On the right, you have a terrible encoding of current because it really is not distinguishing currents. Just it is more like a threshold type, if you like. Although with all the caveats. And this is the plot that I presented in the first class as well for you, that we haven't discussed in detail. But now we can look at it and notice all of the different types of neurons that we now have. Not only class 1 and class 2 excitable standard ones, as we saw just in the slides before, G and H, but all of these other kinds of funny neurons. Right, so going back to the idea of what is a threshold, and then what is causing all of this variability, or all of these different categories. Allow me to note the relationships between the current traces and the voltage traces above. I'm going only to point out to a certain extra few of these ones. For instance, you have a spike latency, you give a current early and the spike happens late. We saw that behave before. In J, you have some sort of subthreshold oscillations. You give a spike, and after the spike, the neuron still wobbles a bit. You have a resonator that's typical class 2 excitable too, where depending on the interval between two currents, even though they have the same amplitude, you might or might not have spikes. So if the neuron is a resonator and has J-like subthreshold oscillations, then it tends to respond like in K, where you produce a spike only if you have a certain delay interval. That's great for measuring intervals, of course. You have an integrator neuron to contrast with that. Integrator neurons, L, are like G, class 1 excitables. So if you have two current pulses close by, you add those currents because of the tau of the membrane, whereas if they're further apart, they don't. Then you have the M, rebound spike, that's also typical class 2 excitable. N, you have a rebound burst, not only a single spike, but multiple spikes. That's also typical class 2 excitable, like chattering neurons in C as well. And you have so-called threshold variability. I mean, it's similar than the resonator in that you give a current, but if you give the same current later, but having primed it with a negative current, you get a spike. So you see, by pushing it down, it actually goes back up. So the idea in O is that you increase the driving force by pushing it down. To see this in a picture, we have to see this in a picture. We have to get a better understanding of why these different spike shapes come to be. And in P, a lovely memory neuron, as it has been called, is the bistability. A single pulse starts a regular firing, a single pulse stops the regular firing. So this gives you a sense of all kinds of dynamical behaviors that even a simple neuron can have. In this case, I haven't talked about the equations on the right yet. So this is a two-dimensional equation, not four-dimensional like Hodgkin and Huxley, but two-dimensional equation that is a simplified model that is being designed to capture most of the, actually all known type of firing types in neurons. So the Zhikovich model. In the project, you're going to be working with the adaptive exponential model, which is of the same sort. And it turns out that all of those firing behaviors can be subsumed in a dynamical systems view of what the neuron is doing. So here, we haven't talked about bifurcations yet, and I'm going to get that in a moment, but allow me to just present you with the fact that you can distinguish all of these kinds of neuron firing types as a function of the different kinds of bifurcations that they undergo. So in this table, you have the names of the bifurcations: saddle node bifurcation, saddle node and invariant circle, supercritical Andronov-Hopf, and supercritical Andronov-Hopf. This material is advanced, and we discuss it in our master's course. But here I want you to simply get a flavor and I'm going to give you a little flavor of bifurcation theory, but I'm not going to discuss them in detail. But ultimately, you have here class 1 excitability and class 2 excitability as being across particular directions of these bifurcations. So if you have here, for instance, an integrator neuron, you don't have subthreshold oscillation, so you don't have resonating behavior. You have a settle node bifurcation if the neuron is bistable, if you can actually turn it on and off, or if it is monostable, you have a settle node and an invariant circle. Don't worry too much about these concepts now. I'm going to get to them in a split second. I'm going to have an iPad moment soon where we're going to talk about the neurodynamics. That comes next. So here we have, in real life, the difference between resonators and integrators. So the basic two classes, they give us quite a bit of information about what is going on in the brain itself. So when we open the brain and we cut a slice and we look into the neuron, we now have good understanding of which neuron applies to which category of brain function. So here on the left, to give you a sense of how these categories matter. Let's start with the integrators on the right. So integrators are very sensitive to input amplitudes. They have a fairly linear response, and they're phasic. They respond when they're given input, and they're quiescent, silent, if they're not given input. So they're great for sensory inputs. So if someone touches your skin, those neurons are silent until you touch them, right? And these neurons then in your brain that are responding to sensory inputs, they're also of that sort. The stronger you squeeze, the higher the firing rate. Great way to encode actually sensory inputs. They're great for associating things, because if you are integrating stimulus from different directions, they perform an AND function. So they also can integrate information. Frequency of input will relate to frequency of output. So that's not only linear encoding, but you can also work as a PID, integrator differentiator type. You actually had some linear systems before, right? And so you have a linear encoding in a certain range, at least for integrators. So integrators are the neurons that we all think about when we think about neurons. You give current, you spike more. And they are the ones that are used for machine learning. So we generally don't talk about resonators at all. So there's a whole new component for machine learning that no one has touched thus far. Neurons that have more of an intrinsic behavior. So it has stiff frequencies, they're tonic, they oscillate, they can be bistable. You toggle it on and off, it stays, it has spontaneous activity. So they are fundamental for autonomic functions. And they central pattern generators, that's what CPG stands for. So if you want to step left and right, you might want that process to be self-driven. And to get self-driven processes, you would like to have a resonator, a neuron that you can toggle. So like, now you're going, now you're going, now you're not. Now you're going, now you're not. The resonators are fundamental in the synchrony across brain areas. When you have electroencephalograms, and you know, maybe you have heard in nanobiology, we don't talk a lot about brain, but if two areas are in phase with each other, that often relates to the fact that these neurons have different resonating properties, can have a phase locking. Resonator neurons are the ones that create timely communication across brain areas. Also for gated transmissions like resonances. So if you want to get input through, say for example, you're doing something, you don't want to, let me see, something that requires attention. Say, for example, you're on the operating table, and you're a doctor, and a bee stings you in the ass. But you are there with the scalpel. You don't want to make a mistake. So you basically totally focused on your hand, and you might not even have noticed the bee that stung you. Terrible, terrible metaphor. But the idea is that the sensory system often is able to blank out and eclipse certain sets of information when you're concentrating on doing something else, when it gets less important. Say, for example, you have pain that you learn to cope with, for example. All right, so you gate that transmission. You feel less pain from certain inputs. There's a lot of examples in systems neurobiology for this kind of difference. And then, because of these resonating capabilities, they can also filter out certain aspects of input stimulus. So this is just to give you a flavor of what these dynamical system differences of the neurons actually impact on when we're discussing what the abilities of the neural systems endowed with these spike neurons could be. Any questions? Because now I'm going to go into neurodynamics. 48, yeah, we're doing okay. If you have no further questions, and if we're good for time, I want to. I'm actually picking up my iPad with my just checking if everything works now. The idea now is that we open the box a little bit about what causes these differences in neurons. The project will also go a little bit more in detail to give you a little bit more intuition about how these dynamical transitions come to be. And I want to introduce you to the vocabulary of neurodynamics so that we can, when I talk about neurons, I can use these words and you can come back to, and there will be a glossary for you to try to parse what I'm talking about. These terms, they are specific, but they're supposed to create a description at a level of dynamical interactions, mathematical vocabulary. So basically what I'm telling you is that you're getting a dictionary from me before we discuss the actual spike generation mechanisms in those terms. Because this is a preview. Neurons are dynamical systems. I'm persuaded this is one of the best descriptions of how a neuron acts as a dynamical system. Resting states of neurons are stable equilibria. The tonic spiking state corresponds to a limit cycle attractor. Neurons are excitable because the equilibrium is near a bifurcation. You inject current, you change the qualitative behavior of the neuron. There are many ionic mechanisms of spike generation. There's sodium 10 types. There's IH, there's calcium-based currents, there is chloride-based currents. We haven't talked about many of them. I mean, like most of them are active. GABA, AMPA, like a whole feast of different conductances. And yet, only 4 general bifurcations of equilibrium, which create all of the categories of neuronal firing as we know. That's mind-boggling. I mean, the fact that we can simplify all of neuronal behavior into 4 mathematical abstract categories. These bifurcations then divide the neurons into integrators, resonators, monostable or bistable, as I just showed you, but just keep it in mind. What we're going to look into here fleetingly is the analysis of phase portraits at bifurcations that explain why some neurons have well-defined thresholds, all or non-spikes, in the case that they do or not, well-defined thresholds, post-inhibitory frequencies, frequency preferences, hysteresis, which is bistability, and so on. These features are not ionic currents per se, determine the neuronal responses, the kind of computation neurons do. We're going to get into this a bit. This is fun. You know, biologists often get infatuated with the discoveries of different ionic conductances in different neurons. Many neurons share ionic conductances, but discovering an ionic conductance is not necessarily a step to understand what the dynamics of the neuron will be, surprisingly. And so ultimately a good model must reproduce not only the electrophysiology but also the bifurcation dynamics of the neurons to explain where they can be with different parameterizations. More words. This slide gives you the overview of the dynamical systems lingo, so the terminology. It will be here in the slides for you to check again. So, state variables, as I explained in the beginning today, are variables that fully define the state of a system. What is a state? A state is a possible configuration of state variables. It's a place where your abstraction describes your reality. So the state is the selection, the state variables are the selected variables that you think are sufficient to describe the state of the system. Sufficient, necessary and sufficient, in fact. The phase space is the space of all possible configurations. So if you have 4 state variables, then your phase space is 4-dimensional. If you have 2 state variables, it's 2-dimensional, and so forth. If you have 4 dimensions in the Hodgkin Huxley, you have, you know, a tesseract, a four-dimensional cube with voltage M, N, and H. In this, if you put a point in that geometrical space, you actually fully determine the system. A trajectory is a sequence of states. So the way in which M, N, and H, V, and change is. The flow is all of the possible trajectories. If you start from this membrane resting potential or this, these are both part of the flow. Equilibria are the loci in the state space of the system that have 0 gradient. So basically, when you are in equilibrium, you don't change your position. I'm sure that you know this from basic physics. A tractor is a stable equilibrium, and a repeller is an unstable equilibrium. A saddle is an equilibrium that attracts from some direction and repels from others. A bifurcation is a change in the state of equilibrium, and I'm going to give examples now in the iPad of these. And the transient is the trajectory before the steady state is reached. Limit cycles are cyclic trajectories towards which the states flow. Most of these terms I have already used in this lecture. Limit cycles only sparingly, but they're perhaps the most important for you to remember. A spike, in fact, is a limit cycle, especially a regular spike is a limit cycle. Okay, now I'm going to transition to my iPad for a moment. And I wanted to remind ourselves of this equation to start with. So the balance of currents in our circuit. Let's start our analysis. Here we have a 4-dimensional system, Hodgkin-Huxley type, but I wanted to start our analysis by simplifying the system a bit and thinking about it, so thinking about only the leak current to start with, if you don't mind. So if you have the leak current here, you have CV dot representing the VDT equals a current that is injected, a parameter, minus the leak of that current. L is missing here. You're gonna love this. I'm sorry, I just noticed that this is too big. Apologies. So L, we'll have to correct this later. So it's effectively a function of V, and you have a couple of parameters, the conductance for the leak and the reversal potential of the leak, right? But if you're willing to simplify a bit, math style, you can take these two to just make it easy for you to handle the equation itself. You might assume, for example, that G is 1 and EL is 0. Without loss of generality, you can include them later, and in which case your V dot equation is going to be equal to an I minus V. And we can even simplify a little bit further. Just let's start assuming that our initial applied current is 0. So V dot equals minus V. That's simple, right? That's really simple. It actually could also be maybe an M dot, which is equal to our M infinity at V minus M divided by tau of M. If you assume for a moment that we're going to 0 and this is 1, this is the same equation. It's nice. It's another way to look into the differential equations, more qualitative way. So if you have this description, you can think about the phase space of the one-dimensional system of this equation here, V dot, minus V, in the space, in the one-dimensional space here indicated by V. So your initial condition, your membrane potential, can start at any V. Your phase space is one-dimensional, so we're running over the V dimension here. So it's one-dimensional, okay? And your state could, for example, be this one. This could be your original state, V0. And then we can ask the question, what is the trajectory? Where will V0 go? So if we now have this system with initial condition V0 equals a certain value in this one-dimensional space, where will it go? Now, this is neat. We can add another dimension here, the dimension of change, and to know how actually, as a function of where we are, what we're going to be doing. If V dot equals to minus V, we have here the identity function. I had already drawn this line which represents f of V. So V dot equals f of V. Now, this is the catch. If you start with a certain V0 here, then the change that you will see in V, you can compute, is going to be related to V0. So your actual speed is a positive value. It's going to be this. It's going to be, if it's on the diagonal, it's going to be the same V0. And then we know that if you start at a certain position, yeah, so I see that Niels Bluesper talked about neglecting the conductance C. Yes, I neglected the conductance C. C is 1. Yes, thank you. Your V is going to have a positive change in this one-dimensional space. That positive change is going to get smaller and smaller as you get closer to the origin. But when you get here, where V is 0, you have no dynamics anymore. So this particular equation is such that if you take the resting potential away from 0 in the negative direction, so if V0 is negative, you have a positive deflection pushing the state to the right. And vice versa, if you now have other V0 starting over here, it can be anywhere, right? So V0s can be anywhere. And you will then have a negative deflection, which will create a flow towards the same equilibrium state at 0. Could be V0 if we had a constant still. This guy here is the attractor of the one-dimensional system. States are going towards it in one dimensions. So if you have now any function, any one-dimensional function that is continuous, you can actually, I'm drawing in the same space, but allow me to have a different function here, a funnier function. I just drew it. It's some other function, f2 of v, because this is f1 of v. This is some other function. Then this function has the action that in these domains here, where the function is positive, initial states are going to be converging into these points. So we will have these attractors, and we'll be moving away from these other points here. Sorry for the somewhat messy. So the negative domains of the function push the function to this attractor. The positive domain push it to this attractor. So this function has an attractor here and a repeller over here, and another attractor over here. Okay, so this is how one-dimensional the dynamical systems work like. If you had your V function, the original one we had, let me actually go back to the past. Back. Eesh, yeah, this is going to be far. Okay, allow me to do it differently. So if we have a thing V dot V, but now we just to complement. So if V dot is has some parameter on V, then this system is the set of all lines with all possible orientations that A can give, as you can probably see, and you will have attractors for certain values and repellers for other values. So for instance, if you have here this one we had before, now I'm going to change color, with a negative A, if A is negative, then we have a line over here, and your states are attracted to the repeller. But if A is positive, you actually have the opposite behavior. So I should actually go here. A is positive, you have something like this, and then states are moving faster and faster away from this attractor. This is in one dimension. And in one dimension, so you can, in continuous systems, this basically captures everything that can happen, and you can quickly visualize it in your mind. It takes a little experience to see quickly the domains of attraction and repulsion, but once you do, this is a great way to visualize any differential equation. In fact, much earlier than solving the equation most of the times, even if it's simply an exponential. In this case, will be exponential solutions, of course. So let us go for one-dimensional systems once again, and let me talk about this particular very useful cubic shape for an f of x. So f of x being v of t in panel A. So we have the one-dimensional state space. We have then these attraction and repulsion domains. Now think about what happens over time. The trajectories of states starting possible different initial states here, where my cursor is marking, they will converge onto the attractors with different speeds, but you know where they're going to go. Okay, so this trajectory, the flow is the set of all of these trajectories. A bifurcation happens if, for example, you have here an f of x plus a certain I here representing current, f of x representing maybe sodium channels. This is a very interesting curve because it actually has attractors that disappear and appear when you have different current values. So when current is 0 for this function f of x plus I, the f dot is simply f of x. But if I is positive, we are raising the baseline of that function. In my iPad, I would probably be doing so. If I have here in the iPad, and I sum a certain I, oh, that's an ugly color, plus I, then I is going to shift these functions, thereby changing the position of the attractor smoothly, right? So it's just basically a constant baseline shift of the function. And here you have a constant baseline shift of this cubic function that disappears with the attractors. If you look at the point where I'm pointing here, and you imagine, I'm talking about the curve that is tangential to the state space, where the value of f of x here is 0. If f of x here is 0, then there is no flow. So this is an equilibrium, and it's still a state, is still a stable equilibrium, because trajectories from the right side is going to be oh, sorry. Now it's no longer a stable equilibrium, because trajectories from the right side will be attracted to this stable point, but trajectories from the, I said right side, so from the left side will be attracted, and from the right side will be repelled, right? So what I'm getting at is this. This is a saddle node bifurcation in one dimension. So if this is your f of x equals x dot, x dot takes positive values here and negative values here. When you have your f of x plus a certain I, and I is 0, trajectories are attracted from the right and repelled from the left. But if you have negative current, you have the emergence of two states. One that is attracting, and the other one that is repelling from this side. All right, just to give you an idea of what happens locally close to the saddle node bifurcation. The saddle node bifurcation underlies the disappearance of an equilibrium and creates a certain dynamics. In B, the last panel we're looking at the position of the stable states. And we have projected now on the y dimension, we have the position of the stable states. Whereas in the x dimension, we have our parameter. Now we've changed, it's no longer a phase space, it's a parameter space. On x, you have current. On y, you have the position of the equilibria. And this is showing, as you change continuously the current, you change continuously the position of the equilibrium. So the equilibrium goes a little bit to the left if you raise the current, and to the right if you lower the current, and then it disappears. So the connected lines, by Hodgkin, which nodes attracting and repelling, and towards which direction this happens. So Andre asked me a question that is fundamental, and I'm going to get to it again because it might get more of you here with me. So let me go back to the one-dimensional case with a line simply, okay? So, oh, that's biggie. Okay, Andre, everybody else. So here, our space for this particular function, okay? Which one is attracting, which one is repelling? States attract, so this is now our function, f of v. And this is actually v dot. So this is v dot is the direction of change of the state v0. So what is v dot when v0 is to the left of this system? Well, since the function here is positive, then dv dt here to all the points to the left, to all the points to the left of the equilibrium, your change is positive. Positive change means that the next state is going to be a little bit more than what you had before. So you know, if you want to go, where does V0 go? V0 goes at a place that is V0 plus a certain delta that is moving positively. So here, this amount here that we're adding to V0, our dV dt, is actually positive. So you're adding a little bit. And so that's why your V0 states move here to the right. And here they move to the left. Here's an exercise for all of you. If you now have yet another phase space, the same kind. So this is V dot and this is V. And I draw a certain function. Actually, let me do another one, just for fun. So say it's tangential here, here it crosses, and it's tangential here. And I'm going to call this 1, 2, 1 and 4 are tangential. Can you tell which direction the state is moving just by looking at this curve? Where are the attractors or the repellers? And in fact, some of them are saddle nodes, right? So you have here, the states are doing this. I hope that you can now figure out. Andre, is this clear? So the idea here is that in the positive, did I actually annotate this correctly? I did not annotate it correctly. I'm actually thinking about you as well. So here, the state is moving to the right, here is moving to the left, here is moving to the right, here is moving to the right, here is moving to the right. Can you give me a thumbs up or thumbs down if that doesn't make sense? Okay, great. Okay, so just, you know, contribute like with positives and negatives. So I hope that now this became clear. So we were looking into the difference of change of the state as a function of the state given a certain function that determines it. The relevance is we're really talking about V as membrane potential. Could be an X, could be any function, could be an M as well. So these are one-dimensional dynamical systems that we just discussed. And here as we change and as we shift the baseline of this function, we change the position of these states. So if I now have here a bifurcation whereupon I raise this curve, this now guy becomes a saddle node. You will see that this domain shrinks and disappears. So the position of these equilibria change as you raise current. So these two equilibria, they will merge as we raise this this part of the function here. Okay, I hope that that clarifies. If it doesn't, we can get back to that. I'm going to put a little video on one-dimensional systems and on two-dimensional systems apart from this that is done also systematically to build up to that particular figure we're just looking into. Okay. So here we go. This is the one-dimensional phase space. Welcome. In 2 dimensions, you also need to understand. And I wanted now to invite you. Okay, so first let me actually talk a little bit about two-dimensional systems. So here, two-dimensional systems have flows in 2 dimensions. And so here the phase space is made of 2 dimensions. Back to the iPad quickly. So if now we have a 2-dimensional phase space, that means that you're plotting now x and y as state variables of that system, simply x and y, and that 2 functions determine what is the gradient at any point of this phase space. So if I take this point here, question is, where is it going to go? A 2-dimensional system is determined by 2 equations. So x dot is some function of where the state is, and y dot is some other function of where the state is. It's a gradient. And it can be, any function. Could, for example, be, so x dot could be 0, and then y dot could be 0, in which case no state moves, and every state stays where they are. So basically, put a pebble inside a flat surface, it doesn't go anywhere. Now, if you add certain components to these, then you can, for example, play with an equation here, x dot, y dot. So if x equals minus x, and y, say, for the time being equals 0, what would happen? Well, for an x that is at the positive side of x, for any value here at the positive side of x, we would be going towards the center. Irrespectively of where y is, we would be going towards the center. So it's a nice little vector field. You've seen the vector fields before, I'm sure. And vice versa, right? If you add a little y component and say now we have a y that is equal to minus y, just to play with. I should really use more layers when I'm doing this. If say our y is now minus y, then when y is positive, we're going to be tending to the middle. And when x is positive, we're going to be tending to the middle. When y is positive, we're going to be tending down. And when x is negative, we're going to be tending to the right. So you will then have a vector field that is bringing all trajectories, no matter where they start, into this big attractor of the 2-dimensional system here. Cool. So this is what happens in 2 dimensions. I'm finished with this for the moment. So I want to bring you back here. You need a little bit of feeling for vector fields. I guess that you already played with this, but I have this very lovely demo that enriches, I think, our perception of what 2-dimensional fields do. And we will be analyzing spiking behavior in 2-dimensional fields in a moment via one simplified model. And what I wanted you to do now is come to this site called Falstat/vector. I'm going to put this in the chat. Are we all here? You might have to do some sort of juggling of a juggling of windows there to keep everything at the same time. I'll let you have some time for window management. Now, what this representation here looks, this is a model. It's a model of hyperspace, as you can see clearly, right? And in this model of hyperspace, every dot is a potential trajectory. They're added to the phase space in X and Y. So here we have a two-dimensional phase space. On the X-axis is X, the Y-axis is Y. What you see when you put this site on, you actually don't get this user-defined field. You get, I think, this. So I apologize. So this is the first thing I have to mention. When you come here to setup, go from setup to user-defined field, please. But observe me doing it. So you start here, you come at user-defined field, and this is what you have. These are the field functions. F of x and y, as we just saw, equals x, and f of x and y equals y. So basically, this is the same of what I had in the iPad a moment ago. So the field functions are these two. So in that case, what we have is x dot equals x and y dot equals y. That's the default at the file stat vector thing. Okay, x and y. What I wanted you to do now is to think about functions a bit. The other example I just gave you is what happened to states when we had the inversion of x and y in both dimensions. All of states, no matter where they start in your phase space, the phase space is the two-dimensional space here. We're adding randomically different initial conditions, different possible states to this phase space, and observing as the trajectories are attracted to the mean attractor. Points are also removed randomically. So this is sampling the possible state space. You can also look into a grid, so not the grid. Streamlines. Look into the streamlines for where these are going, and you can see that the dots remain within the grid lines. So for any given initial condition, it will go to this attractor if you have minus x and minus y. And this is my exercise for you now. I have put together 4 different vector fields for you to play with in my in this slide. And these are all linear two-dimensional systems with a single attractor at the origin. So basically, these are systems that must look like this. X dot equals some ax plus by, and y dot is some cx plus dy. A linear system with a stable state at the origin. The challenge is this guy here, the bottom right vector field. Can you find A, B, C, and D, according to the coefficients of the linear system, that produce this vector field in the file stat vector thing? I wanted to give you 5 minutes, maybe 10 minutes max. When there are 2 solutions, we move on, okay? So go there to file stat, go to the vector fields, and establish a vector field that looks exactly like the one we have. I might give you some more difficult exercises for you to do offline. But start here and pump into the chat your favorite solution. When we have 2 people that have done a solution, we move forward. Okay? Questions are welcome. I'll be answering them in the chat. have to install a piece of software. I think you flip the axis with the first example. Yeah, that could be, happens to me. I am, yeah, when I'm talking and I'm doing it at the same time, sometimes I slip like that. I have to, let me see here. Yeah, I did flip X and Y. X was stop and Y was an X. You're absolutely right. I just confirmed. But let me not go there now to remove the slide. So thank you. So we do have two solutions. X is minus Y and Y is minus X. It was easy, right? Now it can get more interesting, of course. I wanted to, so I noticed I need to install a piece of software offline, and I wanted now to create a little break. So for those of you that haven't solved it yet, and you can also play, take the opportunity to look into what happens with functions for which you might have some intuitions. So for example, what happens if you have a parabola, right? Or a parabola minus a certain constant. So these will create bifurcations in these lines. Let's give ourselves 10 minutes of pause, and let's reconvene at 10:30. Let me also write it down here. Play, and let's reconvene at 10:30. Okay, so let's make a little pause. I think that I'm a little bit behind schedule, but it's important where we're all on the same page here. Okay, questions are welcome, and I'm going to be playing with the slides here a bit in the screen. So I'm a bit late. I have a minute. So I wanted to continue on two-dimensional phase spaces because this is very fundamental to what we're going to get at in a moment, which is the dynamics of spiking. So ultimately, as you've already noticed, I've used I as a bifurcation variable, so a parameter in the one-dimensional system to change the position of the attractor. And that is actually what is underlying much of the spiking phenomenon. I'm going to get to that in a short second. So here, for instance, it's a sample of the kinds of things that can happen in two-dimensional spaces. We haven't seen some of this yet. Stable focus, rotating vector field, and an unstable focus. So this is a sampler of the kinds of attractor behaviors that can happen in 2 dimensions. I did not find, so I was trying to install Mathematica in my computer because I noticed that this machine doesn't have it, and it failed. So I will use an internet site to explain you a little bit about what happens in the bifurcation of 2-dimensional systems. You will have more opportunity to do this in the project itself. And so I'm now debating whether I'm going to get into the topic of null lines here. I think I will leave this to a video for us to look into later. So I'll put it up in Brightspace in a moment, and then after we finish the lecture, you can go into the video that talks about null lines. So the null lines are states where the gradient doesn't change in 2 dimensions. But wait for the video to really make sense of it. Now, here we have a little simulator online. Jack Terwilliger, I don't know how to pronounce his name. Terwilliger, he's American, MIT guy, wrote this very neat blog post where we can simulate the behavior of certain neurons according to parameters. Ultimately, he is implementing a two-dimensional neuronal model, in this case, the Izhikevich model, which is a quadratic integrate and fire model. And what we're looking into here is some spiking. These are the parameters of this model. You can actually select different kinds of models here, for instance, a Hodgkin-Huxley model, and change the parameters that you're already familiar with, although in that case, you don't have the four-dimensional phase space to analyze. Just notice that Hodgkin is misspelled here. And so we're looking into this so-called simple model. This simple model is a two-dimensional integrate and fire quadratic model represented with these equations. We're not going to derive the equations. They are fairly intuitive, but ultimately the idea here, and we're going to talk about how to derive them in a moment, but I first wanted to give you a sense of the dynamics that they undergo. We have parameters A, B, and U for the second variable. The second variable is called the refractory variable. It's in Y, recovery variable. And membrane potential is the X variable. And we can change these parameters here. And by changing the parameters, we actually change the specifics of the phase space down here on the bottom. Let me bring the chat back in so that I can see if you're giving me feedback. And for certain changes of parameters, you will see that the entire phase space portrait changes. For example, if we reduce the current here, we have the appearance and disappearance of equilibrium states. So we have two curves, and these are the Nock lines. So the Nock lines I said that I was not going to talk much about them, but just a bit. So the Nock lines are the functions for which dv dt and du dt equals 0. So basically, places where the gradient in one particular dimension is 0. And so they are here. So one is a parabola, and the other one is a line. So symbolized with that black and white are the types of two-dimensional attractors or repellers, so the equilibrium states of this phase space. And as you change current, you rise the parabola. You can also see that in the equations, and you bring spiking about via the emergence of the limit cycle. Now we're looking into a state here that only goes from -80 to -30 millivolts, and so it's not continuing to the right, it's cropped. There's a reason why it's cropped. So this two-dimensional model is what's called a threshold model, and it basically assumes that once you are at the upstroke of the action potential, you're actually going to produce a spike. And this is an assumption, simplifying assumption, but it is not, it's a static threshold, but it has to do with the sodium related upstroke. In this model, we're simplifying all of these parameters. So you see spiking here via limit cycles. Unfortunately, my Mathematica crashed, and that's my Mathematica installation crashed. So I'm using this as a fallback solution. The other one I think is more appropriate. But in any case, here you can select different values for these parameters and observe exactly what are the types of spiking that we've talked about before. We had a regular spiking a moment ago when I changed the C variable, which is actually the resting membrane potential variable. So the reset potential of the spike. you can get it down to a single spike, or indeed ongoing regular spiking. There's no time to go into all of the dynamics that I gave you a flavor of. I just wanted to bring you to a two-dimensional model so that you can get a flavor for how the phase space interacts with the positions of the equilibria and the bifurcations to produce the different kinds of spiking. Okay? Are the red lines in the right below plot the transients? Very good question. So thank you for the question. In fact, they're the trajectories. They include a little bit of the transient, and then they actually go into the so-called the limit cycle itself. The plots that we have in the project that you're going to go through after we finish this also show you limit cycles more closely and in their entirety for adaptive exponentials. There will be more explanations at the level of the project as well, so that you can play with these more knowingly. So for the moment, I'm giving you a flavor, but that's a good question. So the red lines, both the spikes and in the phase space, they're the trajectory. And the transient happens mostly when the trajectory, you know, when you get the bifurcation via here, the current, the initial moment until we get to this limit cycle would be the transient. Thank you for the question. So this is then a way in which you can play with these equations and see the emergence and disappearance of these equilibrium states. So the site is here, but it will also be in our notifications in Brightspace. So in Brightspace, I'm going to put the two videos for the dynamical systems and this, and also the code for mathematical. Okay, so this page is actually richer, so you can also look into other aspects of it we're actually going to get to. Okay, now back to the presentation. Told you something about dynamical systems and one and two-dimensional systems, but the reason for this is because I wanted to tell you how we get from 4 dimensions to 1 dimension via 2 dimensions. So we start from the Hodgkin Huxley and we do something with the conductances, make certain assumptions to allow us to see what this complex 4-dimensional state space looks like in 2 dimensions. So the reason for simplifications of a complex model are many, and you can simplify many complex models into simpler models. So you can, in general, you would like to simplify when introducing details does not have a direct consequence in the desired study. So what is a good model? This goes back to the question in the first lecture. You would also simplify when the computational costs outweigh the benefits. So a complex model has a lot of equations, might be very time-consuming to compute, and especially if you have very many neurons, gains in the computation of a single neuron revert into the number of neurons you can compute. So another reason is you would like to simplify the model for us humans to understand, and this is particularly the neurodynamics angle here in these simplifications that I will introduce, and to define the level of analysis. Say, like, I want to look into what happens if I change the sodium conductance. You can then focus exclusively on the sodium conductance, make the other ones simpler by making assumptions, establishing, making them constant, and so forth. And in neuronal models, what you generally simplify are the state variables as gates, and you can say, okay, so there's too many gates, there are too many ion channels. Can I get fewer ion channels? And you can also simplify the morphology. Always keeping in the back of mind that there are consequences to the simplifications. But so the TLDR for the action potential, the too long didn't read action potential mechanism for the Hodgkin Huxley, is that there are 2 interacting feedbacks. One is a positive feedback that is fast, the sodium, and a slow effect, a slow negative feedback that there are two, right? And one is the M variable, fast, for the sodium, and the slow variables, N and H, they kick in after the membrane potential rises. So when there's an increase of membrane potential via sodium conductance, you also have an increase of potassium conductance. So your N tends to higher values, more proportion of potassium open, which leads to a decrease in V, which is the same effect, if you notice, of what happens in the inactivation variable. So very similarly, you can also see that in the activity curves of the Hodgkin-Huxley model. I think that I actually, let me see. I am missing a plot. I'm missing a plot. Okay, I'm missing a slide. The slide that I wanted to show you is one with the taus of sodium and potassium. You will notice the tau of sodium inactivation variable, that is H, and potassium activation variable N, are very similar. So that means they're moving more or less together as you raise the membrane potential in the Hodgkin-Huxley. And so what you can think about them two as one. So this is one of the strategies for reduction of dimensionality of a four-dimensional system into two dimensions. So strategies for simplification would look like this. You can simply, slash an activation variable. When the contribution of the certain current or certain conductance is small, you can simply get rid of it. When it's way too slow, you can treat it as a parameter. This applies to certain calcium ion channels and certain chloride ion channels. And also, just because this is what you decided to do, you can also simply say, okay, what happens if I remove this one? Fair question. You can make the activation variables instantaneous. So instead of you having also a tau for the M activation variable, because the M activation variable is so fast, you can eliminate that M dot dynamics and transform it into M infinity dynamics by saying that tau is instantaneous, right? And so you eliminate the need to go to steady state. There will be a graph summarizing this. You can also merge activation gates, which is the strategy that I just gave you the intuition for. Now, when you have the two activation gates and move more or less at the same time, you can combine the two time constants into one and make both, say, for example, N and H, another variable, blah, and which then controls both the conductance from sodium and the inactivation the conductance from potassium and the inactivation conductance of sodium simultaneously via one variable only. You can, in our nuclear option, we can, for example, start with the Hodgkin-Huxley and just see what happens if we completely remove our potassium activation variable. Then you can get models that look like a transient sodium current only, like focus on or you can remove inactivation. So like what would happen if sodium would not inactivate? In fact, it's actually relatively simple. The action potential width changes, it gets thicker, but it actually has the same dimensions the same dynamics, same qualitative dynamics. And so we are left with a set of minimal Hodgkin-Huxley based models that from 4 equations, you get 2 state variables at most. And so for instance, here is a model where we've slashed the tau variable for an activation gate. And you can see here what we've done. So this has only 2 dimensions, your N dot, and your M is no longer a function of time. It has no longer t. You see that we substitute M by its steady state. And these are the phase portraits for the spiking characteristics of a model where we make sodium instantaneous. So you can see that in the phase space now is given by this phase portrait on the two-dimensional system is given by the membrane potential versus the potassium activation variable. And what these different plots indicate are different trajectories, possible trajectories of the spike. Now, notice in the two top plots, okay, so the two top plots, none of them has any current injected. What has been done is the following. The initial state has been shifted. The membrane potential has been shifted. Say, for example, a voltage clamp. We would just, boom, we turn on, we put a battery in the cell very quickly, and we shift the voltage potential just a bit from here to here, from the resting state where it was at minus 65 to here, say minus 50 millivolts. When you do that, you experimentally, you add a little bit of voltage, and I'll get to the question that was asked in a moment. You take the state out of its equilibrium, and the NOC lines will determine that there will be a transient trajectory, the spike in this case, that will take the state all the way into its almost limit cycle back into the resting potential. And so the spike that is produced is a single spike. And so this is a discontinuous jump that we had by clamping the voltage, producing the spike and making it go all the way. On the right side, we have the comparison between two trajectories, each with an individual starting point. We have one starting point that is at a relatively low membrane potential, so minus 55, say, and the other one that is the same one we had before, say at minus 40. In one case, we will have a small trajectory that is just produces a small bump in the membrane potential deflection, and a long trajectory that produces the same long transient, which is the spike. So you can see that even for a single set of parameters without any current injection, trajectories will behave differently depending on their initial conditions. So you produce transients, two different transients, one of which we call a spike and the other one we call a subthreshold voltage deflection. Doesn't the difference between H and M contribute to the action potential? Sue Saka asked. I might have mentioned, but swiftly, so I'll say it again. So the difference between M and H does contribute to the action potential, but to shorten it, to make the action potential a little bit thinner. If you make this sodium instantaneous, thereby and removing H, actually, you make the action potential a little bit thicker, because it doesn't, there's only one feedback mechanism now operating on the state. Now notice the bottom plots. In the bottom plots, we undergo so actually in the top plot as well, and I'll notice. So the bottom plot, we undergo a bifurcation that is related to the current change. So here we have a current change of 10, and here we have a current change of 40. And so you see that the shape of this limit cycle, which represents the action potential, is changed. So here we have a slow frequency, here we have a high frequency. So all of these phenomena of spiking are captured by a reduced model already. So there's a number of things that you can already start to look into in terms of explanations. Also in terms of the appearance and disappearance of the equilibria. So here you see where my cursor is pointing that there is no contact between this cubic curve and this exponential curve, the NO line. And the reason and this is basically the disappearance of the equilibrium resting state, which removes the ability of the neuron to settle again. And so it can only settle into the limit cycle. So the characteristics of the phase space or the phase portrait is such that the resting state no longer exists. Hence, you have ongoing spontaneous spiking for that current injection. Same thing on the right side, and even more so. So this explains, for example, the interactions in the sodium instantaneous with potassium model. What are the relationships between this phase portrait and what happens in the spike itself? So you, in the back of your mind, you know that if you start with the trajectory here in dotted line, you have a trajectory as might be a spike itself. And so if you kick your state out of the resting potential by injecting a little bit of current or deflecting the voltage a little bit, you start the trajectory, transient trajectory that produces a spike, but you fall back into a resting state. So you produce one spike and you're silent, right? And this is the portrait that explains that. And we know then the conditions for this. If you now raise current, you will produce a limit cycle and you will get rid of the resting state. Many simplifications of the Hodgkin-Huxley model exist with 2 dimensions. One of the first ones was this by Fitzhugh and Nagumo, who simultaneously came up with this particular circuit. One actually was working as a mathematician, the other one's an electrical engineer. And you know, this is a model, as you might have recognized on top. It's basically the first 2-dimensional simplified model dynamics for a single neuron. It looks much like a radio because neuron is transmitting messages. So this is a Fitzhugh-Nagumo model. It's a cubic model. I wanted to show you this model dynamics via my Mathematica demonstrations, but unfortunately, Mathematica crashed in my computer and doesn't work now. Not to be bothered, we still have the internet site of Jack Terwilliger so that we can look into these dynamics as well. So these are the knock lines that determine the dynamic of the Fitzhugh-Nagumo. More of it in the video that I'm going to add later. Bistability explained according to phase portrait is here. So you have basically the emergence of 2 stable states with the black line with the black filled dots. These are both stable states. So your neuron can basically either settle on one or settle on the other state. This underlines the notion of bistability, but basically this says that you can have a hyperpolarized equilibrium and a depolarized equilibrium. So the intersections between the knock lines determine where the equilibria will be. The Jacobians of at these equilibria will determine their stability. Come back to the videos, you will understand why. So this is the bifurcation. Okay, so this is a bifurcation. When you have here the disappearance of the intersections between the knock lines, you can, for example, have the 2-dimensional equation of the settle node, and this is how a resting state and a settle node collide to disappear. And so you basically, this is the onset of the limit cycle potentially. So just to look locally at what's happening when we have the onset of the limit cycle. And when we talked about the 4 general bifurcations that explain all of the types of neuronal firing, as we saw before, these are the 4 we were talking about. The settle node of the invariant cycle. Now you also understand what are these 2 dimensions. So this is the potential, the membrane potential, and the other one's a recovery variable. Could be n dot, for example, or n activation variable. So these limit cycles then refer to all sorts of phenomena in that big table with the kinds of spikes. here is then the transition along these bifurcations so that you see what happens to the trajectories for along the bifurcation dimension. So here you have the saddle node bifurcation probably being occasioned by an injection of current up until the disappearance of resting point, the limit cycle emergence. You can also have the saddle node bifurcation happening already at the limit cycle, in which case you have a spiking cycle that is very much similar to the spike you had before the bifurcation. You can have the disappearance of an equilibrium cycle from an unstable state in the middle. This is a funny one. So the Hopf bifurcation. The Hopf bifurcation is basically a typical all or non type neuron. You inject a little bit of current, and it doesn't have a smooth growth of the amplitude of the spike, but an abrupt change. So you inject current, inject current, inject current, inject current, boom, suddenly it starts spiking. Type 2 resonator, Hopf bifurcation. D is a supercritical Andronov Hopf bifurcation. That is the kind of resonator that has a small subthreshold oscillation dynamics. So you have a very smooth growth of the action potential at the injection of current. You can see that the cycle, starts at a point, a resting point, starts to grow as a little circle here, like with a filled thick line. It starts to grow more as a trajectory continues. Whoop, 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 whoop. And that one is a smoother growth of the limit cycle, and so the action potentials get higher and higher in amplitude as you increase the current. So just to come back to what we're trying to explain. We're trying to explain these phenomena, and we see these in vivo, and we don't know why and how is it that the neurons produce them. Turns out that if you simplify these models into only two state variables, 2 dimensions, with a handful of parameters, here we have maybe 5-6 parameters in these equations, most of them even immutable for most of these cells. Notice that in the Izhikevich model here, in the V dot or V prime differential equation, the parameters are not even open. They're just fixed. I mean, look, so basically for all of the neurons, it holds that the sodium dynamics type of activity, V dot V square plus V plus a certain constant, is immutable. You just need then 3 parameters, A, B, and, or actually, 2 parameters for the recovery variable A and B, and 2 more parameters for resetting, which is what the resetting variable for the spiking does. V goes to C if we, larger than 30 millivolts, and U, we have an incremental growth of the recovery variable. So 4 parameters, and you can get all of these spiking phenomena through understanding what their phase portraits are across the bifurcations. This is why this is cool. Once you have this, now it's 12 o'clock. Once you have these parameters, you can go into real neurons and try to find whether these parameters are really sufficient to explain the neurons as we have it. So this is a two-dimensional type generalized integrate and fire model, which is basically an adaptive exponential type model. Here we have a depiction in the figure taken from the paper, but it relates to the equation underneath. So this is also two-dimensional model. It's very similar to the quadratic model that I presented above. But this paper actually shows that by tuning on the fly these parameters to a certain neuronal firing rate. So here you have in black in B, these are the spikes from a real neuron. The gray line is a noisy injection, basically noisy current, fluctuating current. The black here are the different spike moments in a raster. And what the experimenters here try to do, they try to set the parameters for this neuron so that you could reproduce the spiking trains of the real neuron in black via this reduced model in red. I have to add, this reduced model is a single compartment model. So without even going into all of the morphological complexities, just by tuning a handful of parameters, we can predict to 90%, I think it was like 98%, the spikes that are going to come in the future. So this is a very satisfying result from a simplification. You don't need a Hodgkin Huxley. In fact, you can have a generalized model with which you can plug in most neuronal firing types, even to the extent that you can get the real firing rates from real neurons inside. These are the neurons that underlie most of the very large scale brain models of spiking brain models we have out there right now, also in the Human Brain Project. So this is to give you a flavor of what we can do when we understand these parameters. Allow me to do a little bit more and then we go through the project, okay? So are you all, so let me take some questions from the up until now. Any questions up until now? So I know that the material is fairly compressed. It's a primer, so you do not expect to understand everything. There will not be a test, but I would wish that you're able to grasp most of the essentials. There will be some material supplementing this offline as well. If there are no more questions, I'll move on. It's similar. We're now talking about the understanding that a similar set, a smaller set of equations can give us quite a little bit of understanding about what's happening at the biological level as well. So the four bifurcations that we discussed before subsume all of the categories of spiking from reality, satisfying result. The model I just presented you was the adaptive exponential integrate and fire. And you can ask, okay, so now you have like, you know, 4 parameters. What happens? What is everything that can happen under these models? And so you do a parameter space analysis. You play with A, B, C, and D parameters and try to discover to which parameters, what does the neuron do? And these colorful plots here on the left, each color indicates a particular type of neuronal firing. Red is resting, blue is a particular kind of spiking, I think regular spiking, green, and so forth and so on. And you see that we are inspecting, in fact, in this case, the parameter, so different slices of the 4-dimensional parameter space. A, B, C, and D are the parameters, so that's not going to get too confused. So we're basically fixing some parameters of C and D, or A, and in the different panels, A, B, C, and D, and E, and F, you have different slices of the parameter space. The different slices are also symbolized on the right side plot here, in which they did a full-fledged dynamical systems analysis to adaptive exponential integrate and fire models to really understand everything that can potentially happen with these models. Neurons in reality. So have this in the back of your mind. We know in a 2-dimensional model everything that can potentially happen. I want to introduce you to this researcher in computational neuroscience. This is Eve Marder, just gained the Kavli Prize for. She did research where she's shown a couple of very fundamental results for neuroscience. For instance, first, she's shown that just by measuring conductances and average, so say for example you want to make a model of cells, you measure the conductances via some voltage clamps of 20 different neurons, and then you want to know, okay, so you will average these out to plug into a meta neuron. She said, well, that doesn't work, so there is no average neuron. She also shown that neurons mix parameters to find the dynamics that they actually want to have. So even though neurons can have different parameters, what they're looking for is a certain firing pattern. And she also shown with Gina Torrigiano that neurons change to remain the same. That is, you take a neuron out of the brain, you put it in a solution, and you start measuring the neuron over days. And the neuron will change their conductances, but their spiking properties will remain the same. So let me just fly you by these homeostatic results here. So the idea is that proteins are changing, expression is changing, conductances are changing, but the neuron is trying to do what it wants to do. So this is the first paper, Failure of Averaging in Construction of a Conductance-Based Neuronal Model in 2001. People figured out that it was not sufficient to do what people were doing. Parameters for models of biological systems are often obtained by averaging over experimental results from a number of different preparations. But that breaks. Average fails because the maximum conductance of the population of one spike bursters lie in a concave region of parameter space that does not contain its mean. Okay, let me show you what that means. Here, if you measure in experiment conductances, parameters A, B, and C, conductance for sodium, potassium, and leak, and you're doing this in, say, 40 neurons. And so these are, you have like your data points. Each data point is a neuron. What we're displaying here is that blue and red are neurons that they live in different parts of the parameter space. And the average between these neurons is going to be neither belonging to set blue nor belonging to set orange. The idea here is that if you do these 40 measurements of these cells, and in fact they have different combinations of parameters, your average cell represents neither. So this is a funny result. I mean, because we're keen to use averages on models, and this is something you have to be aware. The parameter spaces are not convex. That's also shown in these pictures here, and X in the colorful pictures. You see that the points in parameter space with the same kind of behavior are highly nonlinearly distorted. The space is highly nonlinearly distorted. Makes sense now looking at it. Everybody, I think can figure out, of course it doesn't make sense, but in 2001 we still didn't know that. Some people had intuitions, but that's when we published and secured that to also showing it via data. And so this is actually the data that they had. They were showing that neurons that produced spikes, just like the other neurons, had different conductance expressions. So some neurons produce a spike that looked like the other spike with different parameters. Cool results related imminently to dynamical systems analysis. Result number 2. When you put neurons in a culture and you keep them alive for very long, they carry on changing. But the thing that they're trying to do is they're trying to retain certain spiking abilities, some certain spiking profiles. So they're changing their conductance over time to keep the same kind of firing. Say if your neuron is a regular firing neuron, it wants to be a regular firing neuron, and it adapts those properties, those conductances, so that it remains a regularly firing neuron. Only that, as you just saw, neurons can have different combinations of parameters that relate to the same type of firing. Hence, as we're changing over time our parameters, we can fire the same but have different parameters. And finally, this is also true at the network level. So if you have a circuit with multiple neurons, the neurons want to do the same. So here we have the stomatogastric sensory system of the lobster. So this is basically lobster digestion as a model for you. And you have the pyloric system. So you have 3 neuronal populations. It's a simple system, nice to study. So here on the left side of the panel, you see the 3 populations, and you see that they are spiking and oscillating in different phases. You can produce these oscillations via different combinations of individual neuronal parameters. So the parameter thing is not only true for a single neuron, but also for circuits. So you have on the right side, similar network activity from different network properties. You can also look into the high-dimensional parameter spaces of these neurons. I'm not going to go into this slide much, but the whole idea is that this is a way to visualize these high-dimensional parameter spaces. And so I wrap it with the same slide, just by giving these memorable messages. There's no average neuron. Neurons mix to match, so mix parameters to match dynamics, and neurons change to remain the same. So so that you have, as nanobiologists, you should be thinking about how the cell is constantly changing, right? So this machinery is expressing things, but there's a level, an emergent level of dynamics and function that is eminently important for what actually the circuit does. And so these are 2 levels that are related, causally related, even in the case of parameters to dynamics, but they're different levels of analysis. The emergence of the dynamical / phenomenological level is more important at the level of the organism, whereas the parameters level is the cell machinery, DNA expression, conditions that might be context related. There's a wealth of questions in computational neuroscience that relates to this set of conceptions. The neurons are changing parameters to match dynamics across the whole brain, everywhere. But they're trying to do something. They're trying to keep certain response properties unchanged. This is qualitative behavior. These are invariances, as I've written about them earlier. So the idea that these bifurcations maintain the neurons within a certain spectrum of phenomenologically relevant and functional behavior. All right. So this is the variability set of slides for us, homeostasis and variability. So we had a big arc today. It's compressed, primer. First, we talked about VHH. Then we talked about neurodynamics, abstractly, what are neurodynamics. Talked a little bit about neuronal simplifications and lower dimensional models. And then we said, these are the parameter spaces of the neurons. So this was the story arc for today. And having settled that, I think that now we can go to the project. And we will leave the one-dimensional integrate and fire neurons as the first thing we will do for the next lecture. So today then, I'm going to share with you now the project. I mean, we don't have much time, but we're still going to be like for 40 minutes in Discord to give you support. And this project is also like the Hodgkin Huxley is read and execute and think a bit. No, I mean, it's not so that you can have to do it from scratch. And then the first thing for the next lecture is we're going to focus directly in one-dimensional integrate and fire neurons and their networks. And we're going to see a lot of integrate and fire cool behavior in the large spectrum of spiking dynamics. Questions up until now, I'll leave open. And now I'm going to move over to the project. Okay, I'm going to open the project and share with you the link. So keep the questions coming. I'll come back here in a moment. you can then copy this to your this project that I just pumped to the chat, the project. You can then copy your project to your own drive to work by yourself. You can also basically import the IPython notebook if you have a nice Python installation and you want to run it locally. It's also possible. Okay, so if you save that, it's an IPython notebook, and so you can work with it in Jupyter. This is all what I wanted to really say today. It was a lot of material, but I think that it really wraps the whole umbrella of computational neuroscience, particularly in what's interested in the neurodynamics and neurophysiology aspects of it. So you now know 80%, at least as a broadly category, of what we're doing in computational neuroscience. Not in the level of detail, that takes time, but up until this moment. So project, I'm going to open Discord. I'm going to cut the video. You, if you want, you have the moment for another question. If someone wants to come in with feedback, also write to me specifically in the chat, or if you want everybody to hear your suggestion or criticism, also in the general chat, feel free. I'm going to open now Discord and wait for the last question. Could you perhaps share the link to Discord again? I shall do so. Yes, I can give more info about the project. I'm going to do it in a moment. It should be self-explanatory, but I'm going to walk it over with you. So just give me a moment and while I copy here. Okay, project. So the project today is about the dynamics of the adaptive exponential integrated fire neuron. The adaptive exponential integrated fire is a type of two-dimensional neuron that has been published by actually originally Wolfram Gerstner. It's very similar to the Zhikevich model, but it has a very powerful extra. It has a very fast nonlinearity. So the Zhikevich model has a quadratic nonlinearity, and the exponential has an exponential nonlinearity, and that makes it such that the shape of the sodium channel, the shape of the action potential, is closer to the one that you see in reality. Wanted to do a little bit more of the chat. There is something else that I'm not forgetting. And so you're going to be looking into the dynamics of the adaptive exponential integrated fire and look yourself what are the parameters that lead you to achieve it. So we're going to be calculating some of those functions like Fi curves. Those are frequencies against input current for the different neuronal types. So basically, we have gotten from the original papers not only the equations of the neuron, but also a set of parameters that represents all the types of neuronal firing, so that you can look at the parameters with your own eyes and say, oh, this is what causes a neuron to be resonant. This is what causes a neuron to be a rebound spiking neuron. This is a regularly firing neuron. This is a phasic neuron. You can ask questions about why these parameters exist. Many of them have been tuned, intuited, derived from experiment, but they should all relate to the actual implementation of real neurons. So these are attempting to be biologically meaningful adaptive exponential representations with 2 dimensions of this neuron. You can read the equations calmly later. Also ask questions. So ultimately, I here have a function that parameterizes the neurons again according to their names. We can then run the simulation with the different kinds of neurons so that you can see the different kinds of firing according to the different parameter sets. So this gives you the parameter ranges, the parameter domains. plot them according to a phase portrait and in different dimensions. So here you should all pay attention to the labels of the axis labels. I actually mean the project presentation plus report. So I think then I belabor this. Huh? So thank you, Mario. Mario actually didn't mean this project, but the final project. I'll get to that in a moment. Let me do the following. So the project I'm going to discuss with you also in Discord, okay? Via text. I think that's easier. So basically the project is taking one of those papers and either implementing all of those models or explaining what those papers are. Okay, so those are inspirations for what the projects are. So it's basically taking those papers and transforming into a presentation plus a report, or into code plus a presentation. But I'll get to that in Discord as well. It's also specified, should be specified in Brightspace. Yeah, move on to Discord. Any other questions about the project? And so there's a little homework. Let me just, since I am this far, the homework is for exercise 1, for each of the neuron types above, use the code to parameterize the neuron by its name and reason about its behavior. And the exercise 2 is create FI curves. So these are frequency versus current curves for the following neuronal types: regular, fast, and phasic. And then you have to display them in the same axis and legend. The advantage of this is to see how the neurons differ in their parameterizations as they produce different kinds of phenomenological behaviors. Good. There was a lot of talking on my part today. I appreciate your attention. I see that our numbers dropped substantially. About 15 people dropped from the onset. And so I'm going to be moving on to Discord. We can then discuss it over there. Okay? Any other questions? Moving on. Have a good time. Enjoy your Thursday, and I'll see you next Thursday.